Our next speaker is Dr. Emil Kakkis, founder, president, and chief executive officer of Ultragenyx. Dr. Kakkis founded Ultragenyx in 2010, creating a company built around a deep and critical need for meaningful engagement with patients and their caregivers. Dr. Kakkis is a physician scientist and a pioneer in enzyme replacement therapy and other research breakthroughs that have advanced desperately needed treatments for rare and ultra-rare diseases. Prior to Ultragenyx, Dr. Kakkis formed a foundation called the Every Life Foundation for Rare Diseases. The foundation's mission is to empower the rare disease patient community to advocate for impactful, science-driven policy that advances the equitable development of and access to life-saving diagnosis, treatments, and cures. Dr. Kakkis recently published a book focusing on his 30-year journey to help those living with rare disease, and it has been a truly inspirational one. He is going to speak today about how we define and measure disease and how we could utilize scientific breakthroughs to treat many more rare diseases. Dr. Kakkis, it's a pleasure to have you with us today for Research America's National Health Research Forum. I now will turn it over to you.
Thank you for that introduction. I'm very pleased to be here at Research America National Health Research Forum. I'm going to talk today about the work I've been doing to try to develop rare disease drugs and what I've come to learn over these last years, which is our drug development system needs to move into the 21st century. We need to change how we approach the definition and measurement of disease so that we can use the technology we have more effectively, especially all the latest technology. Now, my key message on the next slide is that if we're going to have precision medicine treatments, and we're working on many of them, we need precision measures of disease. Right now, what we're doing is we have the greatest new technology out there to be able to treat more rare disease than ever. And our development paradigm, though, is old and inefficient. We're looking for the clinical definition of disease: feels, functions, survives, which are very imprecise and difficult ways to measure treatments. We don't have precision medicine evaluation tools for disease. We're just trying to use the old school approach. Now, that may work for a lot of reversible diseases, but many treatments for complex rare diseases will never be developed because the kind of measure that you're talking about won't work, and we won't be able to use the precision medicine therapeutics that we've developed. And we have the science to do better, and it's time for a change to start defining and measuring disease differently. Now, I'll try to give you a little more clarity of what I mean by that. On the next slide, I talk about what I think is a new definition for primary disease activity biomarker. This is a precision medicine definition for a precision disease measurement. That is a precise way of measuring the source of disease, the cause of disease. It's a direct measurement of what is the cause of this disease in a patient. And if we're combining that with a treatment that affects the underlying cause, we think we have a powerful way of taking forth the development of precision medicine treatments in a much more effective way. We have good examples of how we've done that with HIV or Hep C viral load in HIV or Hep C infection. Measuring the virus is giving you a powerful tool on how to improve treatment. And we could talk a little more about that and why that model is so powerful. In the rare inborn error disease where I'm from, phenylalanine levels in PKU is also critically important. An example of a enzyme deficiency that leads to a high amino acid level that's toxic. Ammonia levels in urea cycle defects. These are defects that detoxify ammonia. The amount of ammonia is the amount of disease. You can measure it and treat it. These examples are important in terms of how they change our mindset of what the disease is. Is the disease what happens from the disease, or is the disease the source of disease? And this is why we need to start thinking differently. Now, if you go on the next slide, you can see where this comes from in my own experiences. I worked on developing a new therapy for MPS1 in the 1990s. It started off with no money because there's only a couple hundred patients in the United States maybe with this disease. And I survived making an enzyme therapy that worked in dogs. My families raised money. And we were able to get to the clinic, which was a miracle in of itself. And we had a tremendous time getting in, and then a incredibly difficult time finishing the program. And that story, while successful, is I think, a very important lesson on how our development process is hampering our ability to develop treatments. To give you more clarity on that, I'll show you some of the challenges for Aldurazyme on the next slide and its implications for what's called accelerated approval based on biomarkers. Now, when we first started in the clinic, we met with David Feinblum and the team in Sieber, and they agreed to our precision medicine markers. They agreed to liver and spleen size and uric GAG as appropriate measures of the cause of disease. They accelerated the program, got us in the clinic, 10-patient trial. They said that was it. If you could prove that biomarker improvement, they felt this was going to be adequate, and they were very pleased to see such a rare disease getting finally a specific treatment. Now, we did that study, and it was very successful, but when we came back, unfortunately, David Feinblum, the leader of that group, had died of brain cancer between that evaluation. A new review group came in, and they ignored the biomarker results. They said they don't know what they mean. We can't prove what they mean because they've never been used before in a drug development program. The delays in requirement for a randomized control trial resulted in a quadrupling of the cost, and it really ignored the science of biological plausibility. Now, on the next slide, I tell you a little about what that means. It means we spent 4 times more money. The company funding this nearly collapsed. We didn't learn anything new from the placebo-controlled trial. In fact, what we knew was the same, what the dose should be, how it worked. At the same time, because that program required more investment, 2 other enzyme therapies for MPS programs were canceled, and treatments were delayed at least 10 years minimum. Without the biomarkers and ability to prove MPS 1 treatments with biomarkers, no improvements have been developed. I chart all this tragedy, the excitement and tragedy in Saving Ryan. It's an important read for you if you want to see how the drug development system is working and then sometimes not working. Now, to give you a better understanding of why this is important, why do we need to think differently? I want to bring up an example from another disease called adrenoleukodystrophy on the next slide. Adrenoleukodystrophy is a disease which really is striking in its pathology. It's a disease in which there's a metabolic defect causing the white matter of the brain to degenerate. And what's striking about it, and this is an example of a kid who was originally I saw was 7 years old, and that kid was starting to have some visual acuity problems. He was a smart kid, doing great in school, having some visual problems. They did an MRI. His brain looked horrible. And yet he just began having clinical symptoms. And this is one of the fundamental problems we all need to understand. Your brain is very plastic. It wants to maintain function. It will maintain the appearance of good function despite progression of the disease inside the brain. And it finally breaks down at the end where it no longer can compensate, and now you start seeing symptoms. You see the disaster at the end, but the disease was there the whole time. If you measure disease by clinical, you think disease only started later, but actually this disease started way before. And if we try to fix it clinically, once the kid's affected, it's really too late. That's one of the fundamental problems you need to see in these diseases. I'll talk about on the next slide another disease called Phenylketonuria, a very similar situation. In this case, there's an enzyme mutation that interferes with the metabolism of phenylalanine. The Phe level in your blood builds up. Well, it turns out very high Phe levels are toxic to neurons and myelin, and you start hurting your neurons, but your brain can function for a while. But by the time these kids get to several years of age, they'll have severe intellectual deficiency, and it's irreversible. The truth is, you need to treat them when they're a baby so that by the time they're 8, they're not going to have severe intellectual deficiency that's completely irreversible. But how would you do that? How do you treat a baby and then watch them for several years before you figure out, can you run a trial using a clinical endpoint in this design? You can't. It's unethical to run trials that way, and you'll never get it right because you'll never be able to measure intellectual deficiency effective level. Now we're measuring phenylalanine. The lower you get it, the better you are. And there's no way to do this development if you're going to try to use intellectual dysfunction as an endpoint. So it's very important to understand this. This is not just about a biomarker being an easier way. It is the right way. It is the core of the disease, the source of disease, and it's the only way for some diseases like PKU. Now, one way to make this easier for people who are not familiar with biomarkers is and to really understand they're not just compromised. They're not like simple, cheap ways to get around doing clinical trials where you prove things that mean something. It's really about a fundamental change of mindset. Now, I think when you talk about a disease like PKU, they have I call it equivalent to me having defective brakes. Phe levels go up, they can't stop. The Phe levels go up, causes havoc. Eventually, car crashes, brain is broken. And so the question is, if you're trying to evaluate disease by evaluating the brakes, you would fix the brakes. But if you're evaluating car crashes, you're doing something else. The truth is, we have decided that our drug development system is about measuring car crashes. That's the only thing that matters. But yet, measuring car crashes is a very inefficient way, because when you have broken brakes, how long it takes, how often you have a car crash, whatever else is going to happen to you is highly variable. But you can measure the brakes precisely. If you measure the brakes, you fix brake function, you improve the car. Now, on the next slide, I put that forth to you. What kind of drugs do you make? If you're evaluating the brakes, the metabolic defect, or if you're evaluating car crashes. Well, if you're evaluating the brakes, you can say, well, I can tell the brakes are broken. I can measure it very accurately, and I'll figure out how to fix the brakes, prevent the brakes from falling apart, finding alternative ways to brake, perhaps. But if you're evaluating car crashes, you might come up with something like big soft bumpers, so you don't get hurt, or better seatbelts. You're trying to mitigate the problem, you're not solving the problem. So that's a more fundamental question, that if you look at clinical car crashes, you always are mitigating problems at the downstream. If you want to fix the problem, you got to go upstream. And that means using traditional ways of doing development are not going to allow that. And so we need to think differently about how to develop drugs. Now, one of the pieces of this is shown on the next that's really important. If you look at the 16 years in HIV, in the beginning, before accelerated approval, you're getting one drug every couple of years, and then you end up with a surge in all these drugs, and one drug improving in others, other ones made obsolete. The medical evolution process was accelerated. But look what happened with MPS. When we didn't get the biomarker-based approval, we were forced into clinical. Since that time, more than 10 companies have tried to work on improvements, and there's not a single improvement in 19 years. In fact, the patents expired and not a single drug approved, no improvements. That's the difference between a biomarker approval, using a primary disease activity biomarker, and clinical. That's the disaster that we have been creating for ourselves by changing our mindset to believe that feels, function, survive, our clinical endpoints is the only truth. When the truth is treating the underlying disease, that is where we get to where we need to go. Now, the key thing I'll show you on the next slide, very important analysis we did, that if you were able to prove things on these biomarkers, primary disease activity biomarkers, where you do one study, we could develop nearly 4 times as many drugs for the same amount of money. And people talk about the high cost of these drugs. Well, if we can develop 4 times as many drugs, then the cost structure of development will be lower, and the ability then to press on pricing will occur. We can then figure out a drug development system that gets treatments developed at a lower cost and gets more drugs to the patients. This is the kind of change we need at a policy level. So my final slide, precision medicine. We're all excited about it, but precision medicine requires precision measure of disease. We need to improve the efficiency of drug development. The costs are incredibly high, are going to drive costs of drugs higher, and we need to modernize our view. If you measure the source of disease, you're measuring the disease. The clinical outcomes are variable and certainly are important, but in the beginning, you must develop a way to measure the underlying disease and demonstrate improvement. With that enabled, you will get newborn screening and start treating disease very early in life before it's caused irreversible damage. And we must begin the medical evolution of more genetic rare disease treatments. Just to be clear with all of you, I'm not talking about more accelerated approval. What I am talking about is really a change in the way we define disease and the way we develop drugs. It's time for a change, time for the enter of the 21st century. Thank you.